Science is incredibly much fun to do. A life as a scientist never gets dull. You never have quite enough time, but it never gets boring. When I began graduate school, the brain was just beginning to be understood. And in fact, the part of the brain where most happened at just the time that I was a graduate student was the understanding of the visual system. By the time I was ready to have a career of my own, I didn't want to continue work on where many of the important big questions were already answered. And then I realized that unlike the visual system, the auditory system was just beginning to be understood. We are so fantastically good at understanding what the sounds around us mean and knowing where the sounds are coming from that we don't even notice what a feat that is on the part of the brain. So sound propagates toward me at about a foot per millisecond and enters the outer ear, which acts like a funnel that funnels that sound down the ear canal to the eardrum. Then sound has to go through the middle ear. Now, the reason we need an air-filled middle ear is that sound has to go from air to a fluid, which is the fluid of the inner ear. And to get that impedance matching, we have the 3 middle ear ossicles that take the vibrations from the eardrum, which is a large surface area, and they transmit it through the stapes footplate, which has a smaller surface area. So the auditory part of the inner ear is called the cochlea. It sort of looks like a snail, a spiral. And the important cells in the cochlea are the transducing cells, are called hair cells, and they sit in a row from the narrow end at the apex to the fat end, which is called the base. So the hair cells that are in the base of the cochlea transduce the high-frequency sounds. The ones at the narrowest end of the cochlea transduce the lowest frequency sounds, so that in the end we have a tonotopic organization in the transduction of sound. So the hair cells then separate the sounds into their frequency components and report when there's energy at those frequencies. There's a group of neurons that sits in the middle of the spiral of the cochlea that receives the information from hair cells. So each hair cell is providing information to roughly 10 auditory nerve fibers. And the axons of those neurons are bundled together and they form the 8th cranial or auditory nerve. So in our lab, I decided to try to understand the first step in how the brain deals with its acoustic information. I had great fun starting a lab. The way I started my lab was a little bit unusual because initially I didn't have a faculty position, but I did have a little lab, and I also had no money to buy anything. So I had to build a lot of my own first setup. So I love to think of my youthful self marching across the parking lot with a 2x4 over my shoulder as a way to build my Faraday cage. I also had great fun figuring out how to make a vibration isolation table with just little bits and pieces that I collected. So for example, I managed to find myself a table with no top, so just the frame. And then I was able to put on it a piece of plywood. In this plywood, I cut out some holes that allowed the nibs of motorcycle inner tubes to poke through the bottom. And then by putting a lot of weight on top of these 4 motorcycle inner tubes, I was able to, to decrease the vibrations on the top of the table so that I could make recordings. So before each experiment, I would dive down under the table, get my bicycle pump up the inner tubes so that the vibrations from the floor wouldn't be conducted up to the surface of the table. But I was also beginning at a marvelous time in science. So my department had a lot of support. There were people who helped me with electronics. There were people who helped me build things. There were people who helped me with histology, all of which these days departments can't afford. So I didn't have very much money, but I actually did have quite a lot of help. So I feel actually extremely fortunate that I didn't start up with so much money. So I think I had more fun than people have nowadays. Those recordings that I made with fine pipettes were from neurons in a part of the brain called the cochlear nucleus that get information directly from auditory nerve fibers. So we take slices of brain tissue, put them in a dish so that we can get at them, and then take these small electrodes and make electrical recordings from those neurons. We can see how in the network of neurons in the cochlear nucleus, information is changed and thereby processed. Many of the experiments that were done early on involved taking a tone, beep, and measuring how neurons fire action potentials in responses to that. And so auditory nerve fibers have a very characteristic response, but it starts fast and then it slows down. Well, it was clear very early on, in the 60s in fact, that if one recorded from the neurons that get information from auditory nerve fibers, that they can have quite different firing patterns. So instead of firing brrrt, some of them fire only bump, just one single action potential, and others actually just keep on firing. So it's clear that the neurons that are just firing at the onset of a tone are doing a different job than those that are firing while the tone is on. But what I was curious about is what information those neurons were extracting from what they got from the auditory nerve. So octopus cells were first discovered by Kirsten Olsen, a Norwegian anatomist, who saw that there was a group of cells that had dendrites that all came off of the cell body in one direction. All mammals have them, but all mammals have very few of them. So when we began to make recordings, we found that for the first 10 years, we recorded from every cell that was known except for the octopus cells. So then we realized that the peculiarities of the properties of octopus cells are so extreme, they are so fast and so leaky, that every time we recorded from them, we thought we had damaged the cell. Nobody really knows what octopus cells do, but a very interesting possibility is that they fire with great temporal precision at the onset of natural sounds. And the reason that's important is because marking all of the spectral components associated with one source and separating it from the spectral components associated with another source is a very important function. I think as a scientist, one's results almost never stand on their own. But my work, or the work of my colleagues and mine, have led to a real understanding of how the brain goes about getting the information about sound that they need. So what we've learned, and what my lab has contributed to, is to understand how the information flow in the auditory pathway is subdivided into parallel pathways, each of which does a separate job. The bushy cells tell us where sounds are coming from in the horizontal plane. The stellate cells tell us where the formants are in speech. And octopus cells tell us when spectral components start together, and therefore when they're likely to come from a single source. I find that teaching is an absolutely essential part of the scientific enterprise, because experiments don't do anybody any good whatsoever until they're known, and I think that students have to be trained even to begin to think about this kind of thing.